The Golden Mussel project advances into the third phase, sponsored by CTG Brazil, SPIC Brazil, and Tijuana Energia, under their ANEL R&D programs, executed by BioBureau and Hubs. The golden mussel is the most damaging invasive species in Brazilian fresh waters. With fast reproduction, few natural enemies, and uncontrollable population growth, it removes native species, causing massive environmental and financial losses. Since 2005, our researchers have started molecular studies on the species and envisioned an innovative approach of genetic silencing to create a genetically engineered mussel that spreads infertility to collapse the invasive populations. In 2017, a partnership was established with CTG Brazil. In the first phases of the project, we identified reproduction-related genes and compared several mussel populations throughout Brazil to identify genetic diversity. The third phase of the project, executed by BioBureau and Hubs, includes 4 research routes. We have modeled the mussel population dynamics to understand its life cycle and genetic inheritance, confirming the viability of the proposed technology. We identified target genes that can impose infertility, and the genomic mapping of the species is being upgraded to reach a golden standard, making it an international reference. We have developed a method to measure mussel populations and predict their progression using advanced technologies combining satellites, sonar, artificial intelligence, and mathematical simulations. The fourth research objective is a prototype, the first genetically modified adult mussel engineered and grown in a laboratory using a combination of genetic editing, fertilization, and lab growth. To support the research, a state-of-the-art lab is being built to cultivate the animals and, later on, to produce modified mussels in a controlled environment. The next phase in this innovation journey includes a field prototype, advances in biotechnology, development of a device for autonomous environmental monitoring, and reproduction experiments to demonstrate the induced infertility in a setting that simulates the conditions of a reservoir. Our development promotes advanced planning and reduction of maintenance costs in hydroelectric power plants. In the future, it will pave the way for a more efficient management of the invasive mussel and recovery of impacted environments. The solution is at the forefront of technologies for environmental conservation.